Welcome to this tutorial. It's entitled Considerations in High Dimensional Flow Cytometry, especially about the data analysis. My name is Faris Naji. I'm a co-founder of Tercen, an analysis platform for flow cytometry, especially high dimensional. So this is what I'll be covering. Basically, an intro to cytometry. I won't pass or won't spend too much time on that, but I will spend more on the high dimensional opportunities or challenges, especially to do with the complexity of the data. And I want to show how Tercen can make it simpler, and you can analyze your data with Tercen, the software company I founded, how you can do quality control on this high dimensional data, how you can explore your data, especially using these dimension reduction techniques, which I will talk about, tSNE and UMAP, of course, but also how they work, some of the concepts behind them. Clustering, I will go through some clustering algorithms so you understand how to identify population groups. And I will touch upon biomarker discovery process and how to discover biomarkers, and then how to integrate the data, especially in a translational setting where you may have more than flow cytometry data. So it's a journey. And speaking of journeys, on the right here, you see a book from the 1884 from Edwin A. Abbott. It's a critique of the Victorian times, but it's also, what's more important is that it's this journey. A square, which is a two-dimensional object, goes on a journey on the higher dimensions and the lower dimension. And it's called a romance of many dimensions. And I kind of look at it as the same way as what I'm taking you on a journey. You are the square and you're going on a journey on the higher dimensions. Worth checking out. The world is called Flatland, and the book is called The Romance of Many Dimensions. So, cytometry, what is it? It's a powerful technique where you have a flow of cells going past a laser, and these cells have been previously fixated with conjugated antibodies, and you conjugate them with fluorochromes. And as the cell goes past the detector, you can detect characteristics of the shape and or density of these cells, but also what kind of markers or antibodies, when you fixated your antibodies, what kind of markers were on the surface. And these detectors at the back here are the ones who pick up these excitations of these fluorochromes that connected to the antibodies. So basically, you're getting a readout of the markers that are on the surface. You have a signal processor built into the instrument, and you basically get these signals per channel, they're called, and you can look at these channels, and usually in a bi-axial plot, but I'll show you that there are other techniques as well, so just looking at the bi-axial. So it's a very powerful technique, and it's been doing very well, and it's behind, I think, major finds in the immunology world for years. So there is another approach, CyTOF, which is an improved in terms of system for detection, because instead of fluorochromes conjugated to the antibodies, you're now using metal isotopes. And the advantage in that is that fluorochromes, these detectors, when they detect the fluorochromes, the wavelengths that they detect can step on each other. So you can see this spilling over, or these wavelengths, how they overlap, and so that introduces noise into the system. While if you use the metal isotope, you get these sharp detecting wavelengths, or the wavelengths are sharp and they don't walk on each other, and there's no there's much less noise, and therefore you can go up to higher channels, 50 channels, for example. But in the meantime, the flow cytometry world has improved, and there's this spectral flow cytometry, and it's going up to 40 channels. So it's very exciting times with all these parameters coming online. So because of these this advancement, it opens up new research possibilities, because now you can basically characterize your cells with more parameters. and you can do better science effectively. So manual bi-axial gating, which is a traditional approach to look at how these cells and how to identify these cells using these plots that I mentioned. Now this can be problematic because now you have, for example, 50 channels. To the power of 50 is in the billions. Can't do biaxial plots, or you can, but you can't do them for all of them. So you need more sophisticated techniques. And these are new algorithms, especially the multivariate algorithms. That's a branch of statistics that I've been involved with in the last 15 years. And multivariate statistics can handle 2 to the power 50 dimensions, call them dimensions. You need new statistical knowledge, especially the multivariate approaches. There's a bit of new terminology in this higher dimensional space. There is a tool challenge or software challenge, but I see it as an opportunity. That's why I think a person is taking this fresh approach, and I started the company for this. There's no standard approach yet on how to handle high dimensional. I mean, there are standard protocols, but it's still evolving very rapidly. So you're still a pioneer in this, and hopefully I can help in that adventure, and you can also help in the adventure of discovering the best way to do things. So I have to mention that cytometry and single-cell RNA-seq are feeding off each other, because the single-cell RNA-seq also deal with similar dimension issues as the cytometry, and what's happening is an interesting synergy technologies. So terminology. There is just to be clear about terminology, a channel name, for example, can also be called a parameter in the flow cytometry world, or marker from the wet lab community. In stats, it's called a variable, and in the machine language world, it's called a feature. So all pointing to the same thing, but having different words coming from the different communities. A channel has a signal. Sometimes it's called an expression. Sometimes it's called a measurement, channel measurement. And these channels represent parameters. And a parameter is basically the number of dimensions. We mentioned 50 dimensions. And it can be also these parameters, when you perform dimension reduction, they become components. So these are stats words. And then there's bivariate and multivariate, which I touched upon. Bivariate is 2 variables. Multivariate is more than or many variables. And of course, what's a variable? It's a channel, basically. So the event is another word that's used often. The cell basically represents a cell in the flow world. An observation in the stats world, or cases sometimes in the stat world. They will call them observations or cases. Cluster, when you cluster your cells or your population cluster, it's called a population in the flow world. It can also be called a cluster, of course. A class, sometimes called as a class in the machine learning world, or a label in the machine learning world. This is not a label like labeling in the wet lab context, but a label as in giving something a name in the machine learning world. And I use algorithm, and I also use the word operator, because first in the platform I'm going to show you how to do this high dimensional analysis uses operators, which are basically algorithms, such as these multivariate algorithms that I'm going to show you. So the software landscape is quite large, and each software has its advantages and disadvantages. FlowJo is, I guess, the most popular at the moment, with a great plugin architecture that it can capitalize on the OR community. Kaluza has great interface and has both acquisition and data analysis abilities. Etsy Express is a great tool to downsample, but also to analyze. The Discovery Suite it's more like a combination of products, very powerful, high throughput, and high performance products. So I really like what they're trying to do. And they have an ecosystem effectively, and I'm trying to also be part of their ecosystem, where they to be able to take directly from the machine and analyze your results. Cytobank is from the community and has pioneered a lot of the techniques. So Tercen is the platform. It's taken a totally different approach to everybody else, I think. I think a fresh approach, and hopefully will invigorate the area and improve the science with how it's thinking. Then there's the R and the Python and the Java communities, or languages, I should say. They have a rich source of algorithms. You need to know how to program a little bit. But what Tercen has done is capitalize on this, and we can plug in any or Python, just like Flojo, but I think we've done it in a cloud-based way, so you don't have any of the install problems of Flojo running performance issues. So out of all this, these software tools, I think Tercen and the R and the Python world are basically free, and so you can go there and sign up. So first thing to highlight is the garbage that you can create if you don't think ahead before you do experiment. However, I have rescued data even with these issues, but for example, there's a lot of batch variation that could have been controlled, and if you have more variation coming in, it means there's more work to be done on the analysis coming out. So it's not additive, in meaning that if there's trouble it's an exponential consequence for the data, the amount of time you spend on data analysis. However, you can rescue your data using techniques that I will talk about. So interfering factors that could stop you having good results are your optical calibration is not up to the par, detecting your detector sensitivity and your linearity, your compensation levels, your batch effects, and your biological variation. But you can mitigate all this with maintenance and good experimental design. Experimental design can also cover how you control the biological variation with biological controls. I've had 15 years of experience of this, so this is worth spending time at the start. When you look at your data, you will see these interfering factors. That's why I put a big I here, and in Tercen you can. So the analysis workflow is basically, for the high dimensional approaches, you basically remove your doublets, your debris, or your dead cells. You export your file as a CSV file or a tab delimited file. You upload data to the Tercen platform. You downsample if you need to. You transform your data using ACNH, for example. You perform quality control like Flow AI. You overview your data, which I'll show you. It's very interactive Tercen. You normalize your data if you need to because of batch effects, which you will see in these powerful projections that I'll show you. You then start clustering your populations into different groups. You explore your clusters, and you may have to return here and recluster when you explore and you validate. You have a cyclic process here of determining your clusters, and then you find your targets, or your biomarkers maybe, or your population group. So Tercen, it's a fresher process, I mentioned. I think it's a long chain of evolution, and Tercen is, you could liken it to Homo habilis, and it's interactive. It's very much trying to be the next step after Prism and Excel, which were fantastic products and still are. What's going to be the final product, I'm not sure, but maybe Tercen will evolve into the ultimate product, hopefully. So where does Tercen lie? Just to give you a feeling of why it's a bit different. If you had this axis where you had low and high, where low flexibility and high flexibility, and this is flexibility without knowing programming, on the basically y-axis. On the x-axis, if you had high amount of data and low amount of data, you would see that Prism and Excel are very flexible, but they can handle, when I say low data, less than 1 million data points, for example. And then you have the programming languages, which are able to handle much more data, but you need programming knowledge. And therefore, it's in this quadrant here. Tercen is trying to be up here, where you can handle the large data, the high dimensionality, as well as not need to have to program. So what we are doing here in the in the tutorial, I'll be showing how to upload a CSV file, how to create projections, how to look at your data. I'll be using different data sets like the B cells data set, which has a time channel, so as I can show you various events. I'm going to be using a PBMC data set that is good for population clusters from Kirsten Diggins data sets, and another PBMC from for biomarker discovery from the Bowden-Miller lab data set. I want to show you concepts of variables versus observations, and I want to show you how in Tercen you do these projections and these computations. So effectively how to use Tercen. So I will do that right now. I'll click here to go to the Tercen website. You see it's tercen.com up here in the URL. I'm using Google Chrome and I'm in Tercen at the moment. You're looking at a Tercen project. It's called High Dimensional Flow Cytometry Considerations. I put all the necessary files in here for the full tutorial. And a project can be composed of data sets, workflows, and it can be there can be a README with it, which you can read down here, describing the workflows and the different operators that are used or the algorithms that are used inside the workflows. So you can see which type is which depending on the icon, and I will be showing this right now. I'm in a public area, a public project, and a public project is accessible by everyone. So you can sign up yourself immediately during the tutorial or afterwards. You could just sign up and access everything I show you here. So let's look at some concepts and why I think it's a fresh approach. Well, it's fresh because first of all, you have this open platform, and it's you can add any workflow you want. So I'm going to start a workflow. I'm going to do a new workflow. I'm going to call it concepts so you can understand the concepts first, and then everything else will fall into place. When I create a concept or workflow concept, it opens a completely blank workflow. That's why it's white. And I can right click on the canvas, this white canvas, and I can add a step. In this case, it's a table. Table for Tercen is data. So what is it? What data will I use? I'll use a B cell data set that has a time channel in it, which would be important to show you something now. And so I take, it's not so important what data set I take, but it's to show you the concepts. Once I've added the table, it appears as a step here in the workflow, and you can now begin to work on it. You click on the step and you can add a data step, it's called. This is an important, it allows you to do projections. It opens up in this immediately with this view. We call it the crosstab view, and it's inspired by software like what would be a Tableau in the financial world, but for us, we're doing it for the biology or the science world. And when you click here, these are all the factors that you can use for your projections. This was in the data set. You can view them or you can see them as column headers. But you see here I have the measurement, the time channel, the event ID, the file name it came from, the channel name. As I mentioned, Flow Cytometry has these channels, the row ID and the table ID. So I'm going to take measurement since that's what we want to look at. And once I click on it, you see it goes green. That means I can drop it where I'd like to. And I can drop it either as a filter, a label, a color, an error bar, a row concept, a y-axis concept, an x-axis concept, or a column concept. So I'm going to put it as a y-axis concept because that's where I want the data to be drawn. It puts all the measurements from all the channels in this cell. You see it's multiple, and it automatically orders it from lowest to highest. Then I can order them not based on their magnitude, but based on the time that they came. So I can put the time concept either as a row or any of the previous ones I mentioned, or as an x-axis. I'll put it as a column since I want to see the progression of all these measurements through time. You can see now that you have all the channels that have been measured in each of these time points here. And now I can continue. I can squash them up here because there's so many points. So you can see this is all the channels. And I'm going to separate the channels now. And I take the channel, we call them factors here on the left, and I can drag it over and I'll separate and I'll put it as a row concept so as I can separate the different measurements by their respective channel. You can see the CD19, CD21. So you can see all these channels that have been were in this flow data set. Now, if I zoom in a little bit, you'll see sometimes you have 2 points in one cell. That's because at that time point, there was 2 measurements, and they get unique IDs, event IDs. So I can take the event ID annotation in the factor area here and drag it over my projection. I can put it below the time or above the time. But since I want to, it determines the order. So I'll put it below time because I want time to determine the order and the event ID to be underneath. And you can see now the points are separated. So you have basically your data projected in this fashion where you have the channels, which are called the variables on the row, and you have the event IDs projected as columns, and we call those observations to use the stats words or the words that would be useful for you in understanding high dimensional algorithms. So the variables are on the rows and the observations are on the columns. You can color by the actual measurement. So I can use measurement itself to be coloring, and I can go into a heat map mode. So I can look at the data as colors, where red is very strong and green is the middle and blue is the weak. And you can see there is quite a strong forward scatter area, and the forward scatter height is very strong. Side scatters are also, both channels here are very strong compared to the other channels, which are representing markers. So if I wanted to now perform some function or algorithm on this data, I decide an operator. So what projection you decide, it should match with what operator you choose. So I'm going to choose an operator that will perform a transformation, an aSNH transformation. You can change the parameter here to 5 or 15. This is the scaling or cofactor, sometimes it's called scaling parameter. For standard flow, you use a 15, and for CyTOF, you use 5. So I'm putting 15 at the moment, and I can run the operator on this projection. And it's finished now. I can save the workflow up here. I can go up to the workflow. This is what we did already, and I'll rename this step to say it's an aSNH transformation. It's actually a heat map projection as well, but I'll just say aSNH. And I can, after you see that there's a green box here, or green state, meaning that it has been executed, this aSNH, and I can right click and add a new data step. So I can make a new projection. And what's important about the new projection is I can use the same factors as I had before, as you notice all of it, but now I have also a new factor called aSNH, which is the computed or the transformed data. And it's drawn a view where it put aSNH as the y-axis here before we had the measurement, but now we have the transformed values. And if I make a heat map based on these transformed values, using a heat map again, you see that the actual scale has changed. It's still strong. These forward scatters and side scatters are still dominant in terms of magnitude and signal, but you see that the y-axis, before it was in thousands, and now it's in tens. See it here. So we've performed a transformation, but you can basically go on, and I'll just rename this to a heat map maybe, but you can also perform a clustering, a dimension reduction. You can keep adding steps to perform different functions, and you you can make projections and add new steps, and you can build up steps. I can show you previous, like for example, I'll go to a FlowSOM step. You see that you can do a FlowSOM, you can look at the view. FlowSOM I'll be coming to in the next couple of minutes, but FlowSOM is a clustering algorithm. So I'll go back to the explanations here, and I hope you got the basic concepts of Tercen, really. What is downsampling? Downsampling is when you remove events from your data. You can see from, if you remember from my projection that I did in Tercen, we had a lot of events on the column axis. So it's removing measurements so as you can handle. There's two reasons you would do this. You would remove it because you can, if you use a subset of the data, then you will be more performant in some of the algorithms, like tSNE, for example, which is sensitive to the size of your data. But you can also reduce events because, for example, if you did multiple runs and each run was associated with a patient readout, or each run was associated with one patient, you'd like the number of events to be similar across the patient groups. So you want to be able to compare patients, and therefore you use downsampling to equalize all the events across patients. There is a downside to downsampling, no pun intended, but the downside is that you might lose rare populations when you, of course, if you remove data, you might remove the baby with the bathwater, as it were. So I tend not to downsample myself, but I can see why you need it if you want to balance patients. There are different ways of doing it. You can just randomly choose a set of measurements to remove. That's the random uniform distribution, we call it. You can also do density-based, meaning that for more frequently measured values, you would remove those. Think of an histogram. You would focus on the parts of the data that had the higher counts, or the more frequent measurements. And then there's the opposite, where you do it with the inverse density base, where you try to sample your data set more where there's less frequency of the data. So what's good about the last one, the inverse density base, it'll favor, for example, rare populations, if you have these rare populations in your sample. So I won't be going too much into downsampling, but in Tercen we have the downsampling. So what kind of quality control you need to do? So you can, because this is a machine and it's measuring with the laser, it's susceptible to quality issues like flow rate, and sometimes you get surges. Sometimes you also get increases in measurements, signal acquisition, for example, in regions where suddenly the strength is increased. And you might have also sometimes these outliers on the start and the beginning and the end of your readouts. So this is taken care of by the Flow AI operator or package from the OR world. There's also Flow Clean. In Tercen, how does that look like? So I click on Tercen here. This is the workflow that does Flow AI. I've taken a data set to illustrate basically what these quality control problems are. So this data set is similar to the one I showed you before. It's got a time, event, the channels here. So this projection is what the projection I did before. And you see at the end here, you clearly see some surging, surge effects are happening, and a plateau here, a little plateau effect. So Flow AI is the operator, and it's being applied to this projection where you have time observations as the columns and variables as the row, or channels as the row. So it's been executed already. It's green here, and you can look at the output. And the output is cleaned up. It's the same projection, but now it's using a flag filter, or a filter that's been produced by the Flow AI, and this filter kicks out the surge area. So now you see, for the same projection, you don't have the surges. That's as simple as that, how to apply Flow AI in Tercen. So I'll go back here and talk a little bit more about dimension reduction, which is, I think, at the heart of a lot of the high dimensional exploration. If you want to explore your data, the thing about the human or us or scientists, we can look at patterns in the 2-dimension and the 3-dimension, but we're not very good at looking at patterns in the 13-dimension or the 40 or the 50 dimensions if you have 50 channels. So we need to bring these 50 or 40 dimensions back down to 2 dimensions or 3 dimensions, so as the scientists can see interesting patterns associated with the cell populations, for example. So, and of course, if we do this reduction, we don't want any loss of structure, which is a hard call to make or hard requirement to make, because going from 50 dimensions down to 2, you can imagine that this involves a big deformation or of your data. But there are techniques that do a good job of trying to keep the structure, and you can see them here. I put a list, PCA, and I put a year that it was invented. You can see PCA is one of the old favorites. It doesn't perform that well with the flow world, but you can use PCA, maybe not on the marker measurements, but you can use it in other parts of your workflow. But PCA working on marker measurements doesn't work that well. But it's been around since 1901. Then you have this family of tSNEs. I mean, the tSNE was 2008. It was a major evolution, and it was very good at the nonlinear. I will explain that, why it was better at flow cytometry. Then you had approximate tSNE, hierarchical tSNE, you had a fast tSNE, and you have UMAP, which is the new algorithm on the block at the moment, an improvement for dimension reduction. You can see how a lot of these dimension reduction algorithms have been recent in the last 4 years, basically. tSNE has been around, but UMAP now is the one that we recommend. But I will show a little bit of why there was this massive improvement with tSNE and UMAP, and it's to do with this linear versus non-linear concept. So if you had 2 dimensions and you wanted to go and bring it down to 1 dimension, I do this purely to explain how these algorithms work. Of course, we're going from 50 dimensions to 2 dimensions. I'm going to show it just a toy example at 2 dimension going to 1 dimension, but it applies by analogy the higher dimensions. So if you had, for example, 2-dimensional 2 markers like marker 1 and marker 2, and you had population groups, cell groups like A, B, C, and D, and you wanted to go to 1 dimension, you would have to have a line. That's what a 1 dimension is. And this red line here is the projection of the new dimension. So you want to go from the black lines to the red line. That's the algorithm. And you do that because you can handle 1 dimension easier, let's say. And here the line, if you draw this line through these population groups, you can see that if you project A onto the line, A onto the line, and B onto the line, and C onto the line, and D, you could easily separate A, B, C, and D. So this projection, this new axis could retain the separation of all these population groups, A, B, C, and D. However, if the population groups in the 2-dimension were looking like this, A, B, C, and D, and they look like more like a Swiss roll or a circle, if you drew a line through a red line, you wouldn't be able to separate the different population groups because B, C, and D would be all collapsed on itself. But you would be able to separate A, B, and C, A from B, C, and D. But the way to do it is to draw a line, not a straight line, but a curved line through A, B, and C, D, and you would be able to separate the A, B, C, D populations. And if you look at this red line here, it's a nonlinear. So flow cytometry tends to have the population groups like on the one on the right here. So if we're going from a 2D to a 1D, it's good to use a non-linear component, we call it, the new dimension. And that's exactly what tSNE and UMAP do. They are non-linear, and they draw these components that are non-linear, as opposed to PCA, which was a linear one. This is an interesting example done by Nikolai Oskolov, and he basically took the world map to describe how these algorithms can deform your data. So it was also a toy example, more educational, to understand what's happening from going from 50 dimension to 2 dimension. Here he's doing just showing you 2 dimension and 2 dimension to 2 dimension, because he's using a world map with longitude and latitude, and he has different continents here, and each continent has a shape and a position relative to its other continents. And he shows what happens when you use these dimension reduction. Even though he's doing a reduction back to 2 dimensions, it's to show what's the transformation that happens to your data. PCA scores well here because it's cheating, because this is the world map is a linear longitude and latitude problem, so PCA works very well. But it's more interesting here. Nikolai did a comparison of the tSNE, which is on the left here, bottom left, and UMAP, which is bottom right. And in tSNE's case, you can see that South America has moved. First of all, you see that they're all upside down, which is not so important. As long as the continents, the shape of the continents, we call that the local structure, and their relative positions to each other, we call that the global structure, how the continents are located to each other. We call, you can see that it kind of tSNE preserves the local structures, but South America moved now between North America and Africa. So it's moved up. So this here has moved up into the middle. While in UMAP, when it was applied, it didn't deform the data in terms of South America is still there, North America is there. Of course, it's upside down, but it didn't deform the relative positions to each continent, and it also preserved some of the sizes of the continents. In Tercen, we also uploaded the data. I didn't upload Nikolai's data, but something similar. When I click here, I'll go over there. So here is a world map. The data was uploaded that was similar to Nikolai's, but if you look at the world map here, we put in also the continents. I put Antarctica in as well, a red one here in the bottom. And I wanted to see how the different dimension reduction algorithms deformed the world map. So if you look at tSNE, you can see Antarctica has changed position completely in the case of tSNE. Also, the shapes slightly deformed. In UMAP, even though it's upside down, everything is in the right position. You see Antarctica, of course, is up in the top now, but everything North and South America, Africa, Eurasia. So UMAP is much better in terms of keeping the global structures and the local structures here. PCA does, of course, is cheating because the problem is a linear problem. So that's how it looks. And if you want to see if you apply it, I took the PBMC data set from Kirsten Diggins because it has different population groups. And if you want to see, let me just look at the workflow that has that. It's basically the 3 algorithms. Here it is. And this data set has different cell populations, and you can see how tSNE looks like. It's much more spread, as you can see, compared to UMAP. UMAP is tighter. I'll show you. But you can see that tSNE is separating here the different population groups. And this is UMAP. However, UMAP will do something a little bit better in terms of keeping the global and local structures. tSNE also does local, but UMAP does global as well. And then you have PCA, which doesn't perform very well on the PBMC data here. You can see it doesn't. There's a lot of groups here that are on top of each other. Okay, so that's a brief explanation of this dimension reduction algorithms. This is just a summary a little bit to show you that UMAP is the one to choose. It's good with flow cytometry data. It doesn't deform the data globally, as we saw in the world map. Okay, let's talk about cell clustering. It's very important. It's basically the algorithms that I'll be talking about who group cell populations based on their markers. And these populations represent phenotypic populations, and this is exactly what you're wanting to achieve with flow cytometry, is to be able to look at the populations. However, something important to know is that batch effects, which I mentioned at the start, for example, reagents or patients or, well, patient is not technically a batch effect, but a batch effect would be reagents or the different instruments you used, or the days that you ran, or the reagents for fixating the antibodies. So these are batch effects, and they can change the measurement values and affect the clustering. So you may want to think about either normalization or an experimental design that can cater for it. I will talk about a little bit more about this, but it's something to be careful of. So types of clustering, there's quite a lot of clustering. I put down all the types, starting with the traditional bi-axial or manual gating. Started in 1972. It's still going strong, and that's a way of clustering using this biaxial gating. But you have spades, citrus, flow density, phenograph, X-shift, FlowSOM, hSNE, flow grid. I didn't even put all the. There are quite. I think there's another 10 that I could have put on this list. Flow grid, for example, is coming from the single cell RNA-seq, so it's being influenced by the single cell RNA-seq. And you see the explosion in the last couple of years. So which one to take? It's a good question. Lucas Weber from Mark Robinson's lab, or working with that lab in 2016, did a paper, and he looked at 18 clustering methods, and he had 6 data sets. I have them all on Tercen if you want to play with them. They're very interesting data sets because they have different characteristics, and they've all been manually gated as well as automatically clustered. So there's 4 CyTOF data sets and 2 traditional flow cytometry data sets. And he uses this F1 score to score whether there's a perfect match between the manual gating and the automatic clustering, and a 0 if there's a complete mismatch. And here I give just one result for one data set, the 1132 Dimension data set is below here. And the F1 score here is on the y-axis. So the red bar here is the F1 score, and on the x-axis here you have all the clustering algorithms. You can see FlowSOM scores very high in the F1 score, like 76 or something, 0.76. And you also see this green and red bar, which represents the false positives and the false negatives. You can see there some algorithms have quite a lot of false positives. But the FlowSOM score as well. Here, he also checked to see how performant they were in terms of time and how long you had to wait. And he put that on the score, F1 as an x-axis, but also the time it took to run on the y-axis. And for this data set, the 1132 DIM, you see FlowSOM scores very well. So FlowSOM is what we recommend at this stage. So what is FlowSOM? FlowSOM, the sum, it represents the word self-organizing organizing maps. It's a basic neural net, but it's very good at neighborhood classifying groups of, like an image in an image when you're doing face recognition, for example, clustering the pixels into their respective groups. What's interesting about it, it can also show the relationship with these groups, especially, and also how the markers in each group. And the FlowSOM operator that I'm using is the one in the OR community is using from Sophie in Leuven, and she's in Ghent, and she's made her algorithm available. Using FlowSOM, you have to know what clusters, how many clusters you have. You can ask FlowSOM to find this clusters dynamically, but it usually finds it, in my experience, it under clusters a little bit. But you can ask FlowSOM to find its own, find the number of clusters that it thinks is correct. Or you can explicitly say to FlowSOM how many clusters you want to have. And it has this concept of a seed as well, which means that the algorithm, every time you run FlowSOM, you won't get the same clusters because it uses a different seed, but you can set the seed explicitly and it will find the same clusters every time. But I only do that if you want to for publication. It's good to see how it changes. FlowSOM can be done on all the markers or some of the markers. FlowSOM is also can be done on these reduction components I was talking about in my previous slides. So when you do a 4D or a 50 dimension to a 2D dimension, you can use these two or these components that you get out of the UMAP and the tSNEs, but you have to be careful. If you do FlowSOM on those instead of the original markers, you will be missing clusters. So you have to be very careful. I'll show you how this looks in Tercen. So I'm going to Tercen here. You see that is I'm using a data set again from Kirsten Diggins and the Jonathan Ayers lab, and I use it because it has different types of populations. It's PBMC populations, and you see there's a transformation and a FlowSOM is being applied, and I basically use the same projection as I used before for the B cell data that I showed you, and I put the variables. You don't have to put you don't have to include all the channels here for the FlowSOM, but I put them all here as an illustrative purposes, but you can sub-select this if you wanted only to use some of these. And you run the FlowSOM on this, which has already been done, and you basically classify all these cells, which represents these events here, into for example, here FlowSOM has put 7 groups into has identified 7 groups, and you can look at also this what I mentioned, FlowSOM having this ability to show you the relationship of these 7 groups and how the markers are broken down in each of these 7 groups. We call them clusters, of course. That's what the FlowSOM is about. It's building it. Person is building it now. It's running FlowSOM on the and it's showing the this spanning tree. It's called minimum spanning tree, and it allows you to see how the clusters behave hierarchically. So it's quite powerful and straightforward, but you need to know how many clusters you want. You can also run it. I'll just look at another workflow. You can use, for example, once you've done FlowSOM, you can actually do UMAP afterwards. So you do FlowSOM first, and then you do UMAP, and then you can color the UMAP, which is this dimension reduction, by the FlowSOM clusters, which I'm doing here. And so this is a way of looking at the clusters and appraising the clusters, which I will talk a little bit more. But I also have a workflow where you can use UMAP first, and then use FlowSOM, which is the part that I said be careful. If you're going to do that, you're going to find you potentially could lose. So if you look at how I apply, you could potentially not find clusters. But if you look how I applied FlowSOM here, I did them on not the markers, but the actual components, UMAP 1 and UMAP 2 here on the row. So you can do this. The advantage of doing this is that you have a reduced data set that you're actually FlowSOMing on, and the disadvantage is, and that therefore you can find major groups, but you will miss smaller groups. So there's downsides. So you have to be careful. And that's FlowSOM. It's very powerful, and I recommend using it. So how many clusters? So this is a big question, under clustering and over clustering. And it's better to over cluster and then work your way down to find the right number of clusters. And that's what I do. I run FlowSOM with higher numbers, and I see how FlowSOM, what kind of clusters FlowSOM gives with higher numbers, and then I start reducing them. And you look at these clusters and you see how the markers are distributed inside. either using the minimum spanning tree or using these MEM scores, which I will talk about. But you could also use the marker profiles, and you could calculate how much the markers are changing, the CVs or their interquantile range, their median values. You could also check the clusters with bivariate plots, for example. I like this thing called the MEM score, which is from Kirsten Diggins in the Jonathan Ayers lab. And she gives a score of 10, plus 10, 0, and minus 10 on every of the markers. And it can range between this. And what's nice about this is that you can see if it's plus 10, it means it's basically enriched the marker in that cluster. And if it's a minus 10, it's basically depleted in that cluster. And it's a simple equation here. It's based on the median value of that cluster compared to all the other clusters for that marker. So if you have a higher median in one cluster for that marker, it'll have a higher MEM score. But also if it's homogeneous, this IQR is an interquantile range, and it's a way of measuring variance. And so it looks at, and this component here will have a favor more homogeneous markers. So if you have a minus 10, it means that it was depleted, but it was very homogeneous. A plus 10 would also mean it's enriched and very homogeneous. And you can compare your MEM scores of each of these markers per cluster to see what to do. You can even calculate, you can even use PCA on these MEM scores. You can do distance analysis on these MEM scores, which I can show you. But here I'll go to Tercen first to show you these MEM scores. Here I've done FlowSOM with a 90 clusters, and here I've done MEM calculation on those 90 clusters. And then I look at the heat map of these MEM scores. So now you see per marker, you see the 90 on the columns, you have 90 clusters. For each cluster, you have basically this list of MEM scores for all the markers, where red would be enriched, blue would be depleted, from plus to minus 10. White means there's not much compared to how the other clusters are doing. It's not moving very much. It's not enriched or depleted. So I purposely did 90 clusters. You can now see if these clusters make sense. You can do a heat map. It's called a hierarchical clustering heat map. I'll just log scale here, and I'll make the height a little bit smaller that you can see. But what it's doing is taking the previous view, and it's using a hierarchical clustering, which means that it puts the columns that are similar together. And you can see that there's a lot of columns that are very similar together, and it puts also the markers that are similar together. And you can see this block effect that's happening because there's quite a lot of MEM scores that are similar for many of the groups, and therefore we would have an over-clustering scenario. You can see this blockish effect happening here. And if you count the blocks, 1, 2, 3, 4, 5, 6, 7, you see that you could have 7 might be a better number of clusters to use than 90. So I did that. I did 7. That's what's nice about Tercen is you can take a different branch. I calculate, I use FlowSOM at n equals 7. I use the MEM scoring from Kirsten Diggins' lab operator. And then I look at the heat map here. And in the heat map now I have 7 columns. And I have the same number of channels, of course. And I can look at the heat map. If I do a Euclidean, I'll just log the values because it's easier to look at the colors. And you don't see the blocking effect. Well, you could argue that these two are blocks, but I would argue that this CD 1, 2, 3 looks very different between. So you now have used the MEM scores to appraise how many clusters to use in this case. Let me talk a little bit more about other data sets. I've shown the PBMC data set, but I wanted to show you this data set. In this case, the markers are not all the same. Some markers, this is a data set from Lucas Weber, from the Mark Robinson lab. And in here, you can see, or I will show you the data. It's quite sophisticated in terms of experimental design. Here, he imperpusely overclustered to 100. And it's I want to show you these biological effects. So this is the data. It's called PBMC Bodenmiller. I transformed it, and I will just show you the heat map. So I put the channels on the rows, and I put the actual samples on the column here. and the time axis. So the same kind of projection I've been doing throughout this tutorial. And what's interesting here is that you can see these patients. I've put patient information in this view, and you can see that, for example, patient 2, in patient 2, you see that there is a reference or control and a treatment group. And in the reference, this patient has been there's been a treatment perturbation on it. And on the control part of the patient 2, you can see quite a high amount of signal. You can also see patient 3, patient 4, patient 5 looks different. So we see that there are these systematic but important biological variations. And what's great about Lucas Weber's approach is that he can handle this. This is called a pair design in experimental, where each patient is, the actual treatment group is compared within patient first, because each patient has its own variation. So this is a paired two-group comparison. What's also interesting here is the markers are not the same. We have some markers here on the bottom, which would be lineage markers. They are markers associated with the type of cells, while these markers represents their molecular state. You see they're all about phosphorylation or phosphorylated these are. FlowSOM would be used, you would use FlowSOM on the type, like a t-test, but Lucas called it a differential cytometry operator on your state markers to see which markers in those clusters that you've identified with FlowSOM have changed for each patient between the treatment group and the control group. Would identify which of these markers are relevant for this disease. To sum it up, it's basically a diff-site operator. It's a differential. It's like a very sophisticated t-test from Lucas. It's on Tercen. If you need to know more about it, you can contact me. But it can really handle pair designs, which is really good in patch effects. You need to have two groups to compare. Also with translational aspects here, with translational setting or context, and Tercen is pretty good at this type of integration. And here I have an analogy data set that I showed. Using what's interesting here in the workflow that I have on the right is that you have all these readouts. It came from cytokine, came from proliferation and fact sorting, but it also can incorporate RNA-seq. And then you add these different annotations, the marker annotations, to specify what markers you were identifying or using, a sample annotation where you specify what the sample, extra sample information, and then the patient annotation where the clinical information about the patient is added. You can join them all together and you can perform a UMAP on all this data together. So integrating data sets is straightforward in Tercen. This is the overall workflow. You basically run your samples on a cytometer. You use either FlowJo or Kaluza or Venturi one from the powerful Discovery suite, and you convert your readouts into CSVs. You upload it to Tercen. You do your analysis. What's nice about Tercen is that you can have your bioinformatician and your researcher working on the same workflow. The researcher need programming and can also do the analysis, and it can be supervised by the bioinformatician or by the PI. So you then export either the images or the file. So overall, I think you should spend time on experimental design. That really is important for minimizing garbage in. You should get rid of debris, doublets, and dead cells. You should look at your data, which is easy with Tercen because you have all these projections. You should avoid downsampling. Not you don't have to avoid it, but if you have no reason to do it, don't do it. But of course, if you have to equalize between patients, I would. Watch out for batch effects. This can be reagent effects, machine effects. You will see that if you look at your data, you will see batch effects. You should, and it will affect if you don't correct for it or you don't see the batch effects, so you didn't look for it. It might affect your FlowSOM, so you have to check FlowSOM. It will be affected by batch effects, but if you don't, FlowSOM, you should run FlowSOM repetitively, so as you can see how FlowSOM behaves. I mentioned that it's stochastic. It'll change every time because it uses a different seed, but how it changes is important. And one time that you run FlowSOM, you may pick up different clusters that you might be of interest. You can over-clustering is better than under-clustering. MEM scores to appraise your clusters. Use your domain knowledge because that's important. Not all markers are the same. All of these algorithms treat the markers the same. You should also enjoy and explore with Tercen because I think it's a new approach and it allows you to relax a little bit with your data, start playing. And you should contact us and sign up for free at tercen.com. I'd be delighted to see you there. My name is Faris, just to repeat, and you can go to our website and request a session on our website. Delighted that you spent an hour with me. Thanks very much.